Okay, welcome back. From what I understand, probably the work I gave you Monday now may have taken quite a bit of time, depending on how much you remembered from earlier classes and then how much work you put into trying to recall all that stuff and formulate a note for yourself out of it. So, what that means is today is going to be, I think, pretty short and sweet and isn't going to have a lot of additional information you do after you're finished with our lesson. So, where we left off on Monday, I'd asked you to go back and review what you knew about the internal workings of animal and plant cells. That meant reviewing a pretty long list of cell organelles that hopefully you've been introduced to before, and then sort of comparing the difference between what you find in the animal and what you can find in plant cells. Keeping in mind that there's not one kind of animal cell, there's a ton of variety in the different types of cells you can find within animals. And same for the plant. There's just not one kind of plant cell. There's a ton of variety in terms of different cells you will find within one plant and then amongst all the plants combined. Use the gizmo to also get a bit of an introduction to how there are other types of cells. We have other kingdoms beside animals and plants. There's fungus, there's bacteria, there's protists, all of which you'll be introduced to in grade 11 if you go on with your biology. And also review the microscope, which would be the tool we would be using to examine cells if we were in the classroom. And so, of course, you've had to do it through these little virtual simulations for right now. So that's where things were sort of left. What I'm going to add on today is something called cell specialization, which will also mean a little bit of a conversation about what a stem cell is and some stuff related to that. So let's start your note, and we're going to call it. Here, don't need that out of the way. You're on your side, little elephant. Oh, there you go. Okay. So we're going to call this cell specialization. Okay, and like I said, I'll try and keep it pretty short and sweet today, based on what may have been quite a bit of work for you to catch up and remind yourself of what you may or may not have been taught in grade 8. Sorry, I'm just moving things around a little bit. I might have to zoom out to make sure that. Sorry. Okay, so let's start with the idea that for life to begin, and we're going to use humans. I could pick a different life form, but let's go and build on something that's already familiar. So you would have an egg cell. Okay, and egg cells that you get from the female are full of all kinds of organelles. They're very large compared to the counterpart, which is the sperm. And these two things come together and they start a new life form. So I'm going to draw my little sperm cell. These are not to scale because these are egg cells are so much larger than the sperm cell. So this cell type obviously has a few different features. They're pretty simple cells, so I'm just going to point out some of the features because part of the discussion from Monday was to talk about what a cell is made up of is based on its function, what its job is. So one of the most obvious features that I can point out for the sperm cell would be this right here, a flagellum, which is, it looks like a tail and is there to support movement. Whenever you see that, whether it's on a protist or any other kind of cell, it's all about allowing that cell to move quite efficiently. And so it makes sense that it would be here placed on this cell. The other thing you would see a lot of in here would be mitochondria, because this cell requires quite a bit of energy. It has a journey it's going on, and so it has to power itself to be able to cover distances. Mitochondria are the organelle that we associate with creating energy, so that makes sense. You would see that there. What both of these cells have inside them, of course, is a nucleus, and the nucleus contains all the DNA, and that's going to be important to a discussion that comes later. So this plus this and you get one fused cell. Okay, just going to draw it rather plainly. And I'm only going to label the nucleus. Okay, I'm not going to try and draw all the other organelles and features, of which it would be jam-packed full of stuff to obviously help development and get this new life form going. Okay, so when these two together, you have a single cell. And in the nucleus, you have now all the DNA this organism is going to need for future development. So let's just write a few things here to the side about this. So it contains all the DNA coding for the new organism. So you'll learn more about the specifics of DNA in grade 11 if you choose to take it. I'm going to say that a lot because I know a lot of you are going on to biology as a course, and I want you to know that any little bits of missing things or gaps or lack of detail we're doing, it will be redone with you and built upon when you take that course next year. Okay, so DNA contains all of the information to do and build all of the different components and parts of your body. So every single, that's not true. There are a few cells that don't have a nucleus. Let's go with all the cells in the body that have a nucleus, have this exact same code with all of the information needed for anywhere in your body. Okay. Now, all of the cells don't access the same parts of the code. They don't all build the same things, which is what we're working on today. Okay. But that's why if you leave behind a skin cell, for example, I mentioned this before, they just need to have really, I guess, one cell that they successfully extract the DNA from to identify you as an individual different from everybody else on the planet. So this is where we have all the information to build everything that is a component of you. And in a human's case, that would be 46 chromosomes. Now, what's going to happen from here is this cell is going to divide, and you will have 2 cells. Okay, and that will become 4 cells, etc., etc. Okay, this process of division is going to be what Friday's lesson is on. It is an effort to take this cell and make an identical copy of it and continue to make identical copies so that all the cells that come from here have the exact same DNA code, all the exact same instructions in the same way, in the same places, and that gets passed on as this bundle of cells continues to grow and grow and grow. That process of division is called mitosis, and that's what I'll talk to on Friday. Okay, at some point, these cells will start to differentiate or become different from each other and take on specific functions. So way down the line, okay, and I can't tell you, I'm not sure how many divisions. I'd have to look that up, and I'm not holding that in my head right now. At some point, the cells will start to draw on the DNA differently and take on different roles. So through enough divisions, I'm just going to draw it and throw out some examples on these branches. There's more, okay? But eventually, instead of just being all these identical cells that could become anything, haven't really become anything specific yet, they specialize. So let's put here, et cetera, et cetera. Let's go until cells specialize to take on specific roles. And this is where they become the different cell types. So you could have here at one point, maybe I go from just being a general cell that could become anything to a nerve cell, muscle cell, let's put white blood cell. I'm trying to think of familiar ones. Maybe I'll put a fat cell. Okay, these kinds of things. So definitely performing different functions. Now that said, they all have the same DNA code in the nucleus. It's just I'm choosing different parts of those instructions to build a nerve cell specific to being able to communicate, okay, across the body, within the brain, or to other regions of the body. I'm going to need different things than I would with a muscle cell. So, compared to the white blood cell, which function is to destroy invaders, compared to this one, which would be to store resources for the future and draw upon those resources as needed. So, for example, things that might differ here, an organelle that might show up in quite a bit of frequency here compared to here. This one and this one might have a lot of mitochondria for energy, let's say. This one for communication purposes, and then this one for movement purposes. This would probably have a lot of lysosomes because that organelle is associated with destroying, breaking down. This one might have a lot of vacuoles because of that storage component we talked about. So they're differentiated, they are specialized, they have specific roles to take on, and the cells have a different appearance to them, and they have a different makeup in terms of what's being built inside of them. Okay, hopefully if that makes sense. Now, what we call the cells before they specialize and become unique and different from each other, these general cells are called stem cells. So I'm going to go back up my page a little bit and put a little star or asterisk right here along this kind of chain of general divisions. Okay, so cells that have not specialized are called stem, STEM cells. Okay, so what I have described here, you know what I could have added down here? I could have also added sex cells, right? If I'm a female, then at some point I'm going to make some egg cells. If I'm a male, then I'm going to make sperm cells. So the things we started way back here are examples down here. Cells that have specialized to do a specific job. These ones are for reproduction purposes. Okay, so let's go back to this. Because I started with an egg and a sperm and talked about the development from a general cell to specialized cells down this pathway, this is these are embryonic stem cells, meaning they came from the very, very early stages of life, the first few divisions. Okay, so you can get access to stem cells through embryos. There are also still some stem cells within you as a young person, and there will be as an adult. They're not as easy to access. So as an adult, you can make a stem cell donation, and they would access the stem cells that are in your bone marrow. So they drill in, like inside your bones. There is a material in there. There are cells in there which divide and specialize and become blood cells. So you may or may not known before that your bones are the source of new blood cells. So for people who have blood disorders or potentially immune disorders, things that are affecting their blood, they can benefit from a stem cell donation. So the idea would be that if I have a problem, they can basically destroy my cells in my bone marrow that are creating new cells for me or failing to create new cells for me. And then they can, through a donation, put in somebody else's bone marrow, which has stem cells in it. And then I will replenish with that other person's source of stem cells instead of my own. So where they access those is through, and I'll put a link to this so you can see and read more about it if you want, is through the pelvis. So this great big bone that creates your pelvis, they go in through your back and they draw out the bone marrow from that particularly large bone, and they use that as a donation into somebody who they have basically destroyed or eliminated their own source. Okay. So that would be the place where they would gain access to them as an adult. There's a lot of interesting research around doing things with stem cells. So let's add on here why am I talking to you beyond just that blood one about stem cells, and there's some controversy around their use. So let's do a little thing here on stem cell research, and depending on time, this might lead me to talk about a few other issues, okay, related to this. So first of all, what is really what these cells are interesting for in terms of scientific research is the fact that they can become any kind of cell. So like I said before, you have a stem cell. If you can figure out how to do it as a scientist, it has the potential to become any other cell type. So your body does this, and there's a lot of research into how does it do that? How do I take one of these and turn it into a nerve cell? That in itself might be a really interesting avenue of research, because if you have nerve cell damage, these are the cells that don't replace themselves, okay, very well, or potentially not at all. So if I can figure out, if I have access to some of these, how to turn them into these, then I can do things like repair a spinal cord injury, for example. So there would be a lot of research related to figuring out how to take these and turn them into these to potentially treat or cure a whole bunch of disorders or injuries related to this kind of damage. Okay, and then I can extend it into all these other ones. Right now, the donation system I talked about is mostly about accessing people's adult stem cells to the bone marrow to replace stem cells in somebody else's bone marrow to mostly replenish aspects to do with blood supply and immune system and things like that. Okay, so here's where the controversy, well, there could be lots of aspects to that are potentially to some people in conflict with their belief system. But the biggest one is the type of stem cell. So we talked about how you can have ones in adults, and you can have ones from embryos. You can also, there are also stem cells in cord blood, which I'll come back to in a second. So of these three, if I'm an adult and I want to donate my bone marrow and it's going into somebody else's bone marrow, we're all in agreement. There's not a lot of controversy around that, okay? Or if I want to donate for research purposes, not a lot of controversy around that. This one, the embryo one, comes with a lot of debate and a lot of controversy. And the problem here is we're talking about the early stages of life. If I want to access these, then I need to accept that in taking these cells for research, that potential life will not develop. Okay, I can't use embryonic stem cells without knowing that what would have been a life is not going to become one. Okay. So in Canada, there are some guidelines around this. And once again, I didn't hold it in my head, and I apologize for not looking it up beforehand. But if we want to use embryonic stem cells, then we have to do it within so many divisions. So there's a really limited time frame for accessing these cells where we consider that acceptable, or maybe not the whole population, but our laws and our regulations and the agreed upon pieces, we have an acceptable time frame to use these very, very, very early on in the first few divisions, recognizing that that's stopping those embryos from developing into life forms. So there is a way, there are a lot of embryos that are discarded or aren't used, maybe discarded is the wrong word, due to people attempting to get pregnant with fertility treatment and things like that. So there is a way to access embryos. It's just about whether or not, at what point people are comfortable using those for research. So we could have a whole discussion about this stuff in class, but I'm just introducing you to that right now, and you'll have to mull that over for yourself, I guess. Cord blood is once you have a baby, the umbilical cord. Within that, there are some stem cells. So you can do a couple things. You can donate your baby's cord blood like you would donate your adult stem cells for research purposes or for donation purposes. You could also choose to have that frozen, and parents might choose to have cord blood frozen and stored, because let's say that you grow up and something goes wrong. Let's say your liver fails, maybe you have a spinal cord injury, maybe you have a heart defect, something like that. There are stem cells that belong to you stored somewhere here from your cord blood, so your own cells, which is always nice in terms of no chance for rejection. If in the future they figure out how to take stem cells and turn them into these things, then in theory, if you do need a liver, they could take your own cells and grow a new liver for you in the lab and then give you a donation, which was based off of your own DNA code, your own biological material. So we're not there yet. But like I said before, there's a lot of research in trying to figure out how to take this and do this. So we could do a whole host of sort of repair and treatment for people. It would eliminate, obviously, the need for organ donation from other people if we could do this ourselves with our own material. And I'm sure you would have lots of questions, and we could, like I said, really dive into this issue as a class. But as of right now, unless maybe at the end we'll schedule a Google Meet to maybe talk a bit more, that is an interesting area of research. And you could maybe go back and look at how countries go back and forth about in this whole big thing here, when countries have been okay with stem cell research, when they haven't, which ones, limitations on what can be done in a lab, and where they're at. So there's a lot of interesting articles coming out now, even from within our own country, about the progression from here to here, and how people have successfully done some things in the lab, like taken stem cells and turned them into blood vessels and things like that. I might have touched one about that. But we could also, if we had more time, and so if this is an area of interest for you, you can definitely research and see all the avenues and things that are happening in terms of progressing from here to here. And then once you're able to turn this into whatever you want, what you could potentially do with that science, the problems it would solve, maybe the new problems it would create, which would be a whole other bit of discussion. Okay, so I think that's where I'm going to end for now. And because of potentially all the work you did on Monday or following up to Monday, I'm not going to add anything else to this for now. What you'll see attached is just some further information that you might have a chance to look at related to this topic. If you have any questions, obviously, continue to do what you're doing, email, Google Meet, whatever it might be, to help clarify some of this if I haven't related in a way that makes sense. And then where we're going to go to in the next lesson is talking about how these divisions work, okay, the stages and the process of going from one cell to two cells that are genetically identical to each other, which are exactly the same as each other. Okay? All right. Thank you, everyone. And remember that I'm missing you, and I hope that you have a great day.